Hello, welcome to my presentation on antibiotics in cells. The image on here is of my pet Khaleesi. She is a blue-tongued skink. The endomembrane system is set in the eukaryotic cells apart from prokaryotic cells by forming internal compartments, facilitating complex and specialized processes that prokaryotes cannot perform due to their lack of internal membranes. These compartments create distinct biochemical environments for a specific function, whereas in prokaryotes all reactions happen in a common space. An example of increased efficiency is the mitochondrion, which folded during membrane cristae greatly enlarges surface area, offering more sites for enzymes to produce ATP during cellular respiration. The compartmentalization, which separates different cellular activities, allows eukaryotic cells to produce much more energy per unit volume than similar-sized prokaryotic cells, supporting the energy demands of complex cellular functions. A drug target refers to protein or nucleic acid that a drug interacts with to produce its therapeutic effect. Both prokaryotic and eukaryotic cells have two main types of drug targets: enzymes and ions. Enzymes play a role in metabolic processes such as DNA and RNA synthesis. Drugs that target enzymes can inhibit essential cellular activities, impacting the survival and function of both cell types. Some unique structures that serve as specific targets in antibiotics include the peptidoglycan and the nucleic acids, and lipopolysaccarides, also known as LPS. They are unique bacterial structures targeted by antibiotics, comprise the thick peptidoglycan layer. The nucleic acids characteristic of gram-positive bacteria and lipopolysaccarides typically found in gram-negative bacteria. Antibiotics such as the beta-lactams and glycopeptides disrupt bacterial cell wall synthesis or integrity by acting on these structures, as supported by extensive scientific research confirming their clinical relevance and effectiveness. When a person takes an antibiotic, the drug is absorbed through the digestive system. It enters the bloodstream and spreads throughout the body to target infection sites. Antibiotics can act both inside and outside cells, depending on their chemical nature and bacterial location. Many antibiotics do enter cells through passive transport, diffusing through cell membranes, common for lipophilic antibiotics, or active transport carrier mediated uptake, especially for more polar drugs. Based on the specific drug and cell type, once in circulation, antibiotics may enter human cells via active or passive mechanism, aiming to target bacterial cells while generally sparing human cells. However, although human cells are usually unaffected, antibiotics can significantly disrupt the beneficial microbiome, sometimes causing long-lasting effects. Antibiotics effectively combat bacterial infections by exploiting the fundamental differences between bacterial and human cells. They work by targeting bacterial structures and processes that are either absent or markedly different in human cells, thereby maximizing bacterial destruction or inhibition while minimizing harm to the patient. The clinical efficacy of antibiotics is supported by decades of scientific evidence and demonstrating that these drugs successfully cure infections by leveraging the well-characterized distinctions between bacterial and human cellular machinery. Antibiotics effectively target bacteria in the human body by disrupting essential bacterial processes that are either absent or indistinct in human cells, such as a cell wall synthesis and unique aspects of protein and nucleic acid formation, thereby ensuring selective toxicity.